Hey guys, and welcome back to SciXShai. Today we're going to continue our series on lab concepts with spectrophotometry. Spectrophotometry uses the concept electromagnetic spectroscopy. And to understand this concept, we need to take a look at the electromagnetic spectrum. The electromagnetic spectrum goes from gamma rays, X-rays, ultraviolet light, all the way down to radio waves. And the way it works is gamma rays and X-rays have shorter wavelengths and higher energy, while radio waves on the other side have longer wavelengths and low energy. Wavelength is characterized by the Greek letter lambda, and this wavelength is measured in nanometers. And this is going to be important when we look at the different biological molecules that are measured at different wavelengths. Now, the electromagnetic spectrum is found from light. So when you separate light, you can see these different wavelengths and these different parts of the electromagnetic spectrum being seen. In spectrophotometry, and especially in biology, the two sections of the electromagnetic spectrum that we're mainly going to be looking at is the ultraviolet and the visible light. Spectrophotometry is measuring the amount of light passing through a solution. And here we have a very basic drawing of what spectrophotometry looks like. So you have a light source, and this light source sends out a certain amount of light, and this passes through our solution and finally gets detected at a detector, and you get an output of a certain absorbance value. But it's a bit more complicated than that. First, you have a monochromator, which is a prism that separates out the light into your electromagnetic spectrum, and then you have a wavelength selector, and that selects the certain wavelength that you want to measure your sample with. Finally, your detector detects the intensity. And so you get two values. You get the intensity before, and you get the intensity after. So that intensity before is IO, and then after is IT. And that's used to measure absorbance. So that's the basic concept of spectrophotometry, and you kind of understand the different parts that are involved in this. But how is this calculated, and what are the uses of this? Well, absorbance, which is the value that is taken out or provided from spectrophotometry, is used in bacterial, nucleic acids, and protein assays. So before we get into their applications, nucleic acids and proteins, well, they use the ultraviolet spectrum, or the ultraviolet wavelengths, to be measured, to measure their absorbance, while bacteria are measured at the visible light. And so when we look at these two, visible light goes from 400 all the way to 700 nanometers, and for bacteria, we measure it at 600 nanometers. And so that's the wavelength that we measure bacterial cells at. Ultraviolet rays, they go from 400 nanometers all the way down to 10 nanometers. But our nucleic acids and proteins, they don't need that much of a range. Nucleic acids are measured at 260 nanometers, and proteins are measured at 280 nanometers. And usually when you do these assays, sometimes you'll get a 260 over 280 value, and that is the absorbance of nucleic acids over the absorbance of proteins. And that's sometimes used in your experiments. To better understand how absorbance is found, we can take a look at the beer-lambert law. And so to start off, you have log IO, and if we remember, IO is the value before it goes into the solution over I, and this is generally IT. I is also acceptable, and that is after. And that gives you your absorbance. Now, absorbance also has another equation. You have epsilon, another Greek letter, is the molar absorbance constant. And you can see that this is the units for that. You have the length of the sample or path, that's found in centimeters. And then you have a solution concentration. And this is the beer-lambert law. Generally, when we look at this law, you will get an absorbance value out of this. So mainly we look at the log value. And what we get out of this is we get a graph that shows the different absorbances for these different wavelengths. Mainly, you'll see the absorbance for the wavelength that you're looking at. So here, let's say we're looking at 260 nanometers. And so you can see that you have a high absorbance and you have a certain concentration in your sample. Well, this is used in multiple different ways. First, you can see again the concentration of proteins, or how many proteins are in your sample, just by seeing the absorbance, because proteins absorb that wavelength of light. And so when there's more proteins, there would be more absorbance and less intensity emitted. And when there's less proteins, you would have more intensity emitted because less proteins are absorbing that light. And the same applies to nucleic acids and to bacterial cells. Specifically for bacterial cells, a lot of the times when we're looking at bacterial cells, you want to know at which stage of the bacterial growth cycle they are at. So when we look at the bacterial growth curve, you have seeding, you have growth, and you have cell death. And there's different concentrations or amounts of bacteria that are at each of these stages. And so you want to, when you're doing a study, you want to know at what stage your bacteria are at, to know when you should reculture them. Sometimes certain methods are dependent on the stage at which the bacteria are at. And so the absorbance is used for these types of studies with bacteria. Again, nucleic acids and proteins, you can find the concentration or the amount of protein and nucleic acids within your solution. But overall, when we're looking at this, let's say for bacteria, you can't define if your bacteria is alive or dead. And so that's one limitation of this. And same thing for nucleic acids and proteins. You don't know exactly what nucleic acids or proteins you're looking at unless you've isolated them out before. But for bacteria, nucleic acids and proteins, there's some limitations. Now, aside from absorbance, you also have transmittance, and this is associated with absorbance, so it's not much of a leap. Basically, a transmittance goes from 0 to 100%, and instead of seeing how much is absorbed by your sample, it's how much is allowed to be transmitted. So it goes from 0 to 100%, and this correlates with your absorbance values. So from 0 to 2, you can see that the two scales match. If you notice, 2 is not at 0. It's actually before 0. And if we look at this graph of this is absorbance and this is transmittance, you can see that your 0 is not really met. So you can have values greater than 2 for absorbance. We've talked about the electromagnetic spectrum and how it's used to find the absorbances of different biological molecules or biological matter. So you find for bacteria, you use 600 nanometers, which is in the visible light range. And for nucleic acids and proteins, you use 260 and 280, which are in the ultraviolet ray area of the electromagnetic spectrum. We've also talked about the different parts that are involved in spectrophotometry and how we calculate absorbance, which is using intensity. Also talked about how absorbance and transmittance are related and how you can find one from the other. Well, this wraps up spectrophotometry, and thank you for watching this video.